So the first question I'll be talking about is based on the structure and function of the membrane. And figure 1.1 is a simple diagram of a phospholipid molecule. Explain how the structure of a phospholipid molecule makes it suitable for its function in cell membranes. You may label and annotate figure 1.1 as part of your answer. So this is actually a phospholipid molecule, as you guys know. Okay. So the question asks you to state the structure as well as the function of the specific structure. So this is actually the hydrophilic phosphate head of the phospholipid, and this is the fatty acid tails, which are the hydrophobic region. And the hydrophilic head is actually polar, whereas the fatty acid tails are non-polar. Yeah. So I think for the first question, I'll just explain according to the marking scheme first, and then maybe later I'll ask questions. So, okay. So as I've mentioned, the structure is actually hydrophilic phosphate head and hydrophobic or non-polar fatty acid tails. And then you can also reference to a bilayer because in the membrane, it's actually arranged like this. So it's like it forms a bilayer, okay, but it's closer to each other. Oops. Okay, yeah. And then you can also mention that the tails of the fatty acids are actually saturated or unsaturated. And this actually helps in, so when it's unsaturated, it has a kink so that it can disrupt the closely packing of the fatty acid tails. So by then you can actually decrease the amount of hydrophobic interactions and then therefore making the membrane more fluid, or you can say less viscous. And then I actually mentioned one of the functions already. So it can regulate the fluidity, and then because it's arranged in a phospholipid bilayer, right? That means this part is the hydrophobic region. Therefore, it can act as a barrier to hydrophilic substances. So the only thing that can pass through the membrane directly is the small and uncharged molecules, for example, NH3 and CCl4. And then for the head, since it's polar, it can actually form hydrogen bonds with the water, and then therefore it can stabilize the membrane. So that's done with the first question. This question is also focusing on the structure and functions of the components of the cell membrane. So state two functions of a cell surface membrane other than phospholipid molecules and describe their function. So before I get into the answer scheme, I'll actually tell you that the membrane is made out of three, which are lipids, proteins, and carbohydrates. So lipids, you have phospholipids that we already mentioned, and then cholesterol. So let's explain the structure and function. I mean, the function of the cholesterol right now. So actually the cholesterol is like something like this. And then it binds to a tail here. So it's not straight, therefore it does also disrupt the close packing of the fatty acid tails in the hydrophobic region. This is the hydrophilic region of the cholesterol. Therefore, there will be less hydrophobic interactions, and also it can regulate the membrane fluidity. And also because it contributes to the fluidity, it can actually prevent freezing in low temperature. So yeah, and this hydrophilic region actually does form hydrogen bonds with the water as well, so it can help stabilize the membranes. So there's that. And then let's talk about carbs now. You have glycolipids and glycoproteins. So glycolipids are actually carbohydrate chains that are attached to the phospholipid molecules. And the glycoprotein is carbohydrate chains attached to the protein molecules. And its functions is almost the same. It can be used as an antigen for cell marking and also cell adhesion. And also since it has the carbohydrate chain of the glycoprotein and glycolipid can actually form hydrogen bonds with water. So it does also stabilize the membrane. And for proteins, there are actually a lot of types of proteins, a lot of functions of proteins in the membrane. So for example, there's like the enzymes or electron transport chain, but I think that's kind of hard to get points since you would also require to answer the function part. So I think I would suggest you guys to use like channel protein or carrier protein in answering this question. And so for example, if you want to talk about facilitated diffusion, actually the channel protein and the carrier protein helps in this process, and you actually have to describe. So for example, if you're talking about channel protein, you can mention that it has like a water-filled polar channel that can allow small and charged molecules to pass through. And also for active transport, you can talk about the carrier protein, and you can talk about how it changes shape between the ping and pong to carry ions through from a region of lower concentration to higher concentration. Okay, so that's it for this question. So the second question is also something to be touched on, the structure and function. So linoleic acid is an unsaturated fatty acid that is found in some triglycerides and some phospholipids. Phospholipids are components of cell membranes. Figure 2.1 shows a molecule of linoleic acid. So they mentioned that the linoleic acid is actually unsaturated. That means there are actually double bonds between the carbon atoms here. And they mentioned that it's found in triglycerides, and you know that phospholipid molecules have fatty acid tails. Therefore, this would be in the region here. Okay, so the composition of cell membranes of plants changes in response to changes in temperature. At the start of the cold season, there is an increase in the proportion of phospholipids with unsaturated fatty acids in the chickpea, Cicer aratinum. Chickpea plants that do not make this change do not survive. Suggest how the increase in the proportion of phospholipids with unsaturated fatty acids helps plants such as chickpea survive decrease in temperature. So I think for this question, you kind of have to explain the function of the fatty acid tails here in the membrane. So would anyone like to try in the chat box? Yeah. Or should I call out someone?
Yes.
Okay. Can I have Kirti to try give one of the functions?
I'm sorry, could you repeat the question? It was a bit unclear just now.
Oh, sorry. Can you give one of the functions of the fatty acids that help in this? Sorry, surviving decreases in temperature.
It acts as a fuel.
Sorry?
Fuel?
Fuel?
Yeah, like energy.
Okay. Actually, this one is like more to the structure of membrane. So I guess you have to give the functions in regulating, for example, membrane fluidity. But that's okay. It's a good try. I think we can go on with discussing, and then I'll explain this using one of the slides here. Okay, so since there are double bonds, right, they'll actually form kinks in the fatty acid tails like this one. So as you can tell, when there's a kink, it actually does disrupt the close packing of the fatty acids. So it's like it does occupy much larger space compared to this, when it's just 3 saturated fatty acid chains. So there will be less hydrophobic interactions, therefore the membrane would be more fluid and also you can say less viscous. And it also does help prevent freezing at lower temperature. So with that, I actually answered these 3. And the last marking point says that it can maintain movement of named substances across membranes. So like I've mentioned in the previous question, it's actually arranged in a bilayer form. So this is the hydrophobic region of the membrane or the phospholipids. And then because it can act as a barrier to hydrophilic molecules or substances and prevent them from just crossing directly the phospholipid bilayer. And yeah, that's why it can.
I think it's hydrophobic. Hydrophobic, I think.
Oh, I think I mean it acts as a barrier to prevent hydrophilic substances from passing through. Since it's hydrophobic, it's like that.
You're saying the fatty acid tails prevent or the cholesterol?
The fatty acid tails.
Okay.
Yeah, the hydrophobic region. Yeah, okay. So this next question is actually touching on cell signaling. So platelets metabolize linoleic acid to produce a molecule known as thromboxane. Thromboxane is released by platelets when blood loss occurs. Thromboxane acts on smooth muscle cells in a wall of arteries. This causes arteries to constrict, which reduces blood flow. Explain why the constriction of arteries following blood loss is an example of cell signaling. So overall, what you need to know is that cell signaling actually has 5 steps. So the first step would be secretion. Sorry. The second step is transportation. Third is reception. Fourth, transduction. And fifth, response. So I think you can use these 5 steps as a guideline in answering this question. And as you guys know, in a cell signaling process, you would actually need a cell signaling molecule or a messenger molecule. And then you will also need a target cell for it to act on, as well as a specific response. So you have to mention that the thromboxane is actually a cell signaling molecule. And you can tell by this part, thromboxane is released by the platelets when blood loss occurs. So secretion is done by the platelets. They secrete thromboxane, which are the cell signaling molecule. And second step is transportation. So it should be released into the blood plasma or the blood. So it can be transported to its target cells via the bloodstream. So there's the first and second points. And I've mentioned it has to be transported to its target cells. So you have to mention what is the target cell here. So they did say that thromboxane acts on smooth muscle cells. So the smooth muscle cells are actually the target cells. And then third step is reception. Therefore, it actually binds to the receptors on the smooth muscle cells. So whenever you're talking about binding or like binding to receptors, you would usually think about binding site. And when you think about binding site, you would think about specificity. So you can talk about how thromboxane is actually complementary to the receptors. And then transduction, since it's only a 4-mark question, I don't think they actually need you to be writing into detail for that, but I'll explain that further after this part. 6 is the response. So in order for a response to be carried out, the target cells actually have to do something to carry out this response. So the response that they mentioned here is the arteries that will constrict. And in order for the arteries to constrict, the target cells, which is the smooth muscle cells here, have to contract. Therefore, you have to add this point in, but you cannot write smooth muscle constricts because it's blood vessels that constrict and not the muscle cells that constrict. The muscle cells contracts and relaxes instead. Yeah. And then for the transduction, I'll actually be using this diagram here to explain. So what happens is that when a signal molecule binds to the cell surface receptor, it actually activates this G protein here. And then this G protein will go and activate this enzyme to make second messengers. And then these messengers would go and activate more enzymes to make more messengers. Therefore, it is a series of cascading reactions. And the purpose of this is to...
Hang on. It does not make more messenger. There's only one messenger. The rest are not messengers. Thank you.
Sorry. Yeah, it causes a series of cascading reactions. And this is to amplify the signal, actually. Yeah, so there is this part. So this question is actually on osmosis. Okay, so figure 5.1 is a photomicrograph of human blood cells that have been placed in a solution of sodium chloride. So state the function of cell labeled Y. Will anyone like to try this question? I guess you can write the name of cell Y and also the function of cell Y if anyone would want to type it in the chat. Okay, I think I should call somebody. Joyce, would you mind answering this question?
Is it phagocyte?
Yeah. Do you know what cell Y is?
Cell Y.
This one.
Do I have to name the cell?
If you know, but it's not in the question, it's just for knowledge.
Or is it lymphocyte?
It's
Lymphocyte?
neutrophils actually, but yeah,
Oh.
specifically neutrophils, and you can tell it's neutrophils because of its multi-lobe nucleus and also the granular cytoplasm. So neutrophils are actually phagocytes. So yeah, the function is to phagocytose dead cells or pathogens. And you can actually also write endocytosis, but it's accepted because endocytosis is actually it's separated into two different types, which is phagocytosis, which is a cell eating process, and pinocytosis, which is a cell drinking process. So if you were to write pinocytosis, it's definitely wrong, but if you write endocytosis, it's kind of correct. But I think if you know for sure that it's phagocytosis, then it's better for you to write that. So yeah, and then the next question. The red blood cell labeled in figure 5.1 is described as crenated because it has an abnormal shriveled appearance. So explain how this red blood cell has become crenated. Can I have Hei answer this question?
Is it because there is a difference in water potential? The water potential surrounding the red blood cell is lower. That's why there is net movement of water out of the red blood cell.
Correct. Okay, so this question is actually talking about the crenated red blood cells, which are seen here. Like these are all the crenated red blood cells. And you all know that crenation actually is a process where excessive water moves out of the cytoplasm of the red blood cell by osmosis, and osmosis only occurs down the water potential gradient. So down water potential gradient. So that would have mean that the external environment, the water potential of the external environment, which is sodium chloride here, has actually lower water potential gradient than the cytoplasm of the red blood cells. So there's this one mark here. And therefore water would leave the red blood cell by osmosis. And I think a good way you can answer these kind of questions is by using this guideline. So what moves from where to where through what by what process, and it's driven by. So for this case here is water moves from the cytoplasm of the red blood cell to the sodium chloride solution through the partially permeable membrane by osmosis down the water potential gradient. Okay, so that's all for this question. The next question is actually touching on facilitated diffusion. So as blood passes through the small intestine, small soluble products of digestion such as glucose are absorbed into the capillaries to be transported to the liver. Figure 4.2 is a transmission electron micrograph of intestinal epithelial cells. At the surface labeled S, so it's here, movement of glucose molecules out of the intestinal epithelial cells occurs by facilitated diffusion. So outline the facilitated diffusion of glucose molecules. Would anyone like to try using this guideline here? Would anyone like to try to answer? I think I'll call someone. Philbert, do you want to answer?
So from the glucose moves from the gut lumen to the capillary through the.
Okay, I think here they didn't really specify the moves from where to where, but do you want to just try, like, what do you think the key points are of this question?
The facilitated diffusion.
Yeah.
So I know it's by facilitated diffusion, which is driven by ATP, but wait, no, facilitated diffusion is through the carrier protein or the core protein, right?
Yeah.
Okay, and then it's the ones that is transported is the glucose molecules, and then moves from where to where is
It just says out of the intestinal epithelial cells.
so also through the cells of the cell surface membrane, is it?
Actually, since it's facilitated diffusion, that means it needs something to aid in this process. That means you actually need a carrier protein or a channel protein. But for glucose, it's carrier protein. And you guys know that carrier protein is actually, it has binding sites. So it has a specific molecule that it binds to. And for this one, it will be glucose. And you can also comment on the specific binding site, how like glucose is complementary to the binding site of this carrier protein. And you cannot say it's pump protein because pump protein usually refers to, like, for example, the sodium potassium pump that is against the concentration gradient. And that is actually active transport, but this is facilitated diffusion, which is down the concentration gradient. So because it's down the concentration gradient, it's passive. Therefore, it does not need energy or ATP. And yeah, I've mentioned movement is down the concentration gradient, or you can also say from high to low concentration. Okay, so the next question is on active transport, or yeah, it's mainly on active transport, but you also can answer in terms of facilitated diffusion. So sodium ions cross cell surface membranes using facilitated diffusion or active transport. Explain why sodium ions cross cell surface membranes by these mechanisms and not by simple diffusion. So does anyone want to try answering in the chat box? If not, I think I will call somebody. Okay, I think ions are too big. Yeah, Pei is actually correct. It's because sodium ions are charged ions. And if it's for a substance to move across a cell surface membrane directly, it actually has to be small and uncharged. But you know that sodium ions are charged or hydrophobic, therefore it cannot pass through the hydrophobic region of the phospholipid bilayer. Like I've mentioned just now, this is the phospholipid bilayer. So this part here actually acts as a barrier to any hydrophilic substances that passes through the membrane. Yeah. So it must pass through a channel protein or a carrier protein, or you can also use transport proteins. So if you're talking about carrier proteins, that would mean it moves from a region of a lower concentration to a higher concentration, which is against the concentration gradient. So that one is for carrier proteins. And if you're talking about channel proteins, it's for facilitated diffusion only. So that one you can say down the concentration gradient, which is here. And the reason it can pass through these channel proteins is actually because they have water-filled polar channels. So this is due to the hydrophilic amino acids lining the channels. So yeah. Okay, I think we're in the last question actually. This question is touching on the diffusion rate of the this question is touching on the diffusion rate subtopic. So a student investigated the effect of changing the surface area to volume ratio on diffusion. Two different size blocks of agar X and Y were made. The agar contained universal indicator solution. Universal indicator solution changes color when acid is added. The blocks were placed in a dilute hydrochloric acid at the same temperature. The student timed how long it took for each block to change color completely. So just for your information, the universal indicator actually turns red because of the hydrochloric acid. So this question is like they'll time how long it takes for the block to change to a red color completely. So first question, the surface area to volume ratio of block X is 5:1. So this is block X. And they're asked to calculate the surface area to volume ratio of block Y, which is this one. So show your working. Can I have Shumin to try calculating? See here. Oh yeah.
So wait for a while, I need to calculate.
Yeah, you can take your time. Calculation has to be in cm.
So the surface area is all of the faces of the cube, the
Block Y.
cuboid. So it's 6 times 3, and 4 faces, that is 6 times 3. So 6 times 3 times 4, and there's 2 faces, that is 3 times 3. So 3 times 3 times 2. This is the surface area. So
So the answer is 90, and the volume.
is 6 times 3 times 3.
And what's your final answer for the ratio?
5 but 3.
Yeah, okay, that's correct. Actually, I think the marking scheme is wrong here for the units. It should be cm squared and cm cubed, since they mentioned here the dimensions are in cm. But yeah, it's correct. The surface area is actually same because for this side are the same. So what she said is just 6 times 3 times 4 and then 3 times 3 times 2 for the top and the bottom. And then the volume is just 6 times 3 times 3. So you put it in a ratio format and you get 1.67 to 1 or 5 to 3 as she mentioned. So the second question, the student observed that block X changed color completely in a much shorter time than block Y. Can Januli answer this question? It's actually a 2-mark question, yeah.
Is it because block X has a higher surface area to volume ratio, so there's a faster rate of diffusion?
Yeah, correct. Okay, so I'll explain this question by Fick's law. So it's actually, it mentions that the diffusion rate is the membrane surface area times the concentration gradient over the diffusion distance. So I'll use L as a symbol. And also, you can already tell since they've given you that the surface area to volume ratio of block X is 5 to 1 and your calculation for block Y is 5 to 3 or 1.67 to 1. So there's that. And also, since the surface area is greater here, that's why the diffusion rate will increase for block X. And this one also mentioned block X has more surface area proportionately per unit volume. That means that for every 1 cm cube of volume, it has 5 cm square of surface area. And for block Y, it's for every 1 cm cube, it has 1.67 cm square of surface area. So yeah, that's what they mean by this part. And it also has a shorter distance for the diffusion to the center. So I think I'll drop this. So for example, the center is right in here. And there's like this part. This distance would be 1.5 cm. And this distance as well will be 1.5 cm. But for this one, in the center, it will be just 0.5 cm away from the diffusion surface. And because this one is small, so the diffusion rate will be greater or higher. So suggest how the results of this investigation help to explain why plants need a transport system. I'll just go straight into the marking scheme. So it mentioned that the distance will be too far to reach all cells and tissue. So for example, if you were to mention diffusion of, say, water from the root hair cells to a cell in the leaf. So for example, and the epidermal cells, okay? That's actually a very long distance. So again, from this equation, when this one, I'll use a different color. When diffusion distance is greater, this one is inversely proportional, therefore it will decrease. So diffusion rate will be too slow since the distance is too far. And when the diffusion rate is too slow, that means more time is required to carry out this process of diffusion. So the time taken will be too long. Yeah. And then the last question. An experiment was carried out by the student to investigate the ability of reducing sugars to diffuse through Visking tubing. Figure 6.2 shows the apparatus used. So at the start of the experiment, the external solution did not contain any reducing sugars. At intervals, the student tested for the presence of reducing sugars, both within the Visking tubing and also the external solution. Yeah. So name the reagent that is used to test for the presence of reducing sugars. Can I have Yin Ting answer this question?
Is it Benedict solution?
Yep, correct. So that's the answer for this. And for reducing sugars, you actually need to carry out the Benedict's test. And just for information, the way to carry out this test is that you collect 2 cm³ of the sample that you want to test, and then you add in an equal volume of the Benedict's reagent or solution. So if you add 2 cm³ of the sample, you take 2 cm³ of the reagent, and then you mix the test tube, and then you let it boil for around 2 to 5 minutes. Then you observe the color change from there. Okay, so yeah, I think that's it for my part of the presentation. I actually did cover as many subtopics as I could. So yeah, I think if you guys have any questions, you can ask right now.
Okay, so just now Megan mentioned about there's one question that says that phospholipid molecule, the phosphate head, the hydrophilic phosphate head is able to form hydrogen bond with water molecules, and that stabilizes the membrane structure. So then you actually need to know which part of the phosphate head is able to form the hydrogen bond. So this one here, no, not this one, sorry. Hang on. Okay, this one here is delta negative. So it can form hydrogen bond with the water molecule where the H is delta positive. Okay, so therefore it can form hydrogen bond with the delta positive water. Right, so this exact part. And of course, the choline molecule, so the choline has got a positively charged N+. So this N+ will also attract water molecules to it, but it will not form hydrogen bond, right? Because it's not a delta negative N. It's not a delta negative N. It's a positively charged N. So it will attract the delta negative O of the water molecule to surround it. Okay. So there are two parts to the phosphate. If you talk about phosphate head, means it's this one only because it's phosphate, right? Then for phosphate head, it can form hydrogen bond with water. Okay. If you talk about the hydrophilic head, then it also includes the choline. All right. Okay. So this is the first one I want to mention. And the second one that is important for you to know is cholesterol. Okay, cholesterol molecule. Now, just now, Megan mentioned cholesterol has got this hydrophilic region as well as the hydrophobic region. Okay, so the hydrophilic region, right? If you know it very clearly, it is actually made of OH. Okay, this OH molecule. So this OH molecule is also able to form hydrogen bond with water because you know that all OHs, they have got a delta negative O, okay, and a delta negative H. So because of that, this OH is able to form hydrogen bond with water. So again, from here, if it can form hydrogen bond with water, it can also help to stabilize the entire structure of the membrane. Okay. And last one I want to mention is for all the glycoprotein and glycolipids, but all the glyco means the carbohydrate chain, they will only be found outside the cell. So this membrane got two surfaces, right? The outer surface and inner surface. So all the carbohydrate chains can only be found at the outer surface of the membrane, not inner. So you've got another carbohydrate chain here. Then it's found at the outer surface. So this carbohydrate chain will also be able to form hydrogen bond with the water molecules because carbohydrate chains are made up of the monomers, which are usually glucose, okay? And you know that glucose, every carbon has got an OH attached to it. Of course, cannot be this OH because this is being used to form the chain, right? But other glucose that are not used to form the chain, other OHs that are not used to form the glycosidic bond, so they can actually form hydrogen bond with the water. And you also need to know the location of the OH. Okay, so all these OHs, they can form hydrogen bond with water molecules. So there'll be a lot of hydrogen bonds that can be formed along the chain with water molecules, maintaining the stability of the membrane structure. Okay.